Welcome to Lesson 17.1, Genes and Variation. In this lesson, we'll define evolution in genetic terms, identify the main sources of genetic variation in a population, and state what determines the number of phenotypes for a trait. Typical plants and animals contain two sets of genes in their cell nuclei, one contributed by each parent. Specific forms of a gene, called alleles, may vary from individual to individual. The particular combination of alleles an individual carries is known as its genotype. An individual's genotype, together with environmental conditions during its lifetime, produces its phenotype. Phenotype includes all physical, physiological, and behavioral characteristics of an organism, such as eye color and height. Why do biological family members resemble one another, yet also look so different? Similarities come from shared genes. Most differences come from gene shuffling during reproduction and from environmental influences. A few differences may be caused by random mutations. Natural selection acts directly on phenotype, not genotype. In other words, natural selection acts on an organism's characteristics, not directly on its alleles. Why do we say that natural selection never acts directly on genes? Because natural selection selects an entire organism, not a single gene or a group of genes, to either survive or reproduce or die without reproducing. In any population, some individuals have phenotypes that are better suited to their environment than are the phenotypes of other individuals. Better suited individuals have higher fitness, produce more offspring, and pass more copies of their genes to the next generation than less fit individuals do. So inherited variation and natural selection result in differential reproductive success of individuals with different phenotypes. So how does natural selection affect genotypes by acting on phenotypes? Genotype largely determines phenotype. Phenotype directly affects how an organism interacts with its environment. Organisms with adaptive phenotypes are more likely to pass their alleles, and therefore their genotypes, on to the next generation. Because members of a population interbreed with one another, they share a common group of genes called a gene pool. A gene pool consists of all genes, including all alleles for each gene, that are present in a population. Researchers study gene pools by examining the number of different alleles they contain. Allele frequency is the number of times an allele occurs in a gene pool, as a percentage of the total occurrence of all alleles for the same gene in that gene pool. For example, in the mouse population shown, the allele frequency of the dominant B allele, which is black fur, is 40%, and the allele frequency for the recessive little b allele, the brown fur, is 60%. The allele frequency of an allele has nothing to do with whether the allele is dominant or recessive. This picture shows a total of 50 alleles. 20 alleles are big B, black, and 30 are little b, brown. How many of each allele would be present in a total of 100 alleles? Well, in a total of 100 alleles, 40 would be black, big B, and 60 would be brown, little b. In this mouse population, the recessive allele occurs more frequently than the dominant allele. Evolution in genetic terms involves any change in the frequency of alleles in a population over time. For example, if the frequency of the big B allele drops to 30%, the population is evolving. It's important to note that populations, not individuals, evolve. Natural selection operates on individual organisms, but the results of natural selection are changes in allele frequencies in populations. So based on this sample population, assuming we have 25 individuals in this population, how many of our mice would be heterozygous black? How many would be homozygous black? And how many would be homozygous brown? 48% of 25 would be 12. 16% of 25 is 4. And the remaining 36% homozygous brown would be 9. So this shows you the distribution of the alleles and the phenotypes that would be expressed. If the current generation of homozygous brown mice had no offspring, how would this affect the allele frequency of the little b allele in the next generation? Well, that frequency would decline. Genetic variation may produce visible variations in phenotype, such as the different colored kernels in these ears of maize. Other kinds of genetic variation, such as resistance to disease, may not be visible, even though they are more important to evolutionary fitness. So, what evolutionary mechanisms are sources of genetic variation? The 3 evolutionary mechanisms that generate genetic variation are mutation, genetic recombination during sexual reproduction, and lateral gene transfer. A mutation is any change in a DNA sequence. So how common are mutations? Researchers suggest that each of us carries roughly 300 mutations that make our DNA different from that of our parents. Most of these mutations are neutral. 1 or 2 are potentially harmful. A few may actually be beneficial. Most heritable differences are due to genetic recombination in sexual reproduction. This is why children do not look exactly like their parents or siblings, even though they all have all their genes. Remember that during meiosis, one member of each chromosome pair is shuffled randomly into each egg or sperm with one member of every other chromosome pair. In humans who have 23 pairs of chromosomes, this can produce 8.4 million gene combinations. Check out this math. The number of different possible combinations of chromosomes in gametes is 2 to the n, where n is the haploid number. And in humans, we have 46 chromosomes. So the haploid number is half of that, which is 23. So which source of variation brings more diversity into a gene pool, mutation or sexual reproduction? Most diversity in a gene pool is due to sexual reproduction, not mutation, because millions of gene combinations can be produced through independent assortment and crossing over. So how many different combinations are possible in a species with a haploid number of 3? Well, that would be 2 to the third, which is 2 times 2 times 2, which is 8. Sometimes genes pass from one individual to another, or even from individuals of one species to another. Passing of genes from one individual to another individual that is not its offspring produces a kind of gene flow called lateral gene transfer. Lateral gene transfer can occur between individuals of the same species or individuals of different species. Genes relate to phenotype in different ways. In some cases, a single gene produces a simple trait. Other times, several genes interact to control a more complex trait. What determines the number of phenotypes for a given trait? The number of phenotypes produced for a trait depends on how many genes control the trait. Traits controlled by a single gene may only have 2 or 3 distinct phenotypes, depending on the number of alleles involved. In the cell species shown, some snails have dark bands on their shells and others don't. In this species of snails, a single gene with 2 alleles controls whether a snail's shell has bands. It's therefore a single gene trait, a trait controlled by only one gene. This graph shows the percentage in one population of snails with bands and snails without bands. The allele for no bands is dominant over the allele for dark bands. All genotypes for this trait have one of 2 phenotypes, bands or no bands. The bar graph shows the relative frequency of phenotypes for this trait in one snail population. It shows that the presence of dark bands may be more common in a population than the absence of bands. This is true even though the allele for shells without bands is the dominant form. So in populations, phenotypic ratios are determined by the frequency of alleles in the population and by whether the alleles are dominant or recessive. Many traits are controlled by two or more genes and are called polygenic traits. Each gene of a polygenic trait often has two or more alleles. As a result, a single polygenic trait often has many more possible genotypes and even more different phenotypes. Often, those phenotypes are not clearly distinct from one another. The graph shows the distribution of phenotypes that would be expected for a trait if many genes contributed to the trait. What does the shape of the graph indicate about height in humans? The shape of the graph indicates that there is a range of heights in the population. Few people are very short or very tall. Most are in the middle range. Height in humans is one example of a polygenic trait. Height varies from very short to very tall and everywhere in between. So let's review. Evolution occurs due to the change of the relative frequency of alleles in a population over time. The alleles in an organism do not change during the organism's life.